So at Arbor this year, I'm going to present our research in Nanophotoswitches, which is a family of molecules that responds to light, and we hope that it can be used towards building a molecule-based retinal prosthesis to restore sight to the blind. We have tested the activity of these molecules with advanced electrophysiological technologies at USC, and we also imaged the fluorescence of these molecules with our multiphoton system at USC too. So in photoreceptor degenerated eyes, the inner retina, namely the retinal bipolar cells or ganglion cells, lost their signal input from the degenerated or deteriorated photoreceptors. Therefore, the visual signal transduction in the retina is lost. So our technology aims to bypass the damaged photoreceptors and directly activate the retinal ganglion cells. So when these molecules are injected into the eye, they will diffuse and embed themselves in the membrane of the retinal ganglion cells. And when under the light exposure, visible light exposure actually, these molecules will be excited and they will either donate or accept electron from the environmental molecules, other molecules in the environment. And when they accept an electron, they will change the membrane potential of the ganglion cells, and thereby inducing spiking activity of the cell types. The advantage of this technology is that since the acting unit is single molecule, it has the potential to achieve very high resolution, very high acuity prosthetic vision compared to what we see in market now. So in market, we have seen bioelectronic retinal prostheses that have restored some level of vision to the blind, but the resolution is typically pretty poor. And using our technology, we hope to finally establish high resolution prosthetic vision in these patients. I cannot give out a time frame at this moment, but we are working really hard towards translating the technology from bench to the clinic. Right now, there are a couple of questions that need to be answered. First, how long these molecules can stay stable and active in the ocular environment. To understand this, we need to study the removal mechanisms of these molecules in the eye. And second, whether we can develop a improved or optimized delivery technology to target these molecules to specific retinal neurons, such that the neuronal signals of this ganglion cells, for example, can better incorporate into the visual signal pathway.